We are going to move to the translational and system genetics session. And our first speaker is Monica Justice from SickKids in Toronto. And she's going to be talking about suppressor screen in Mecp2 mice reveals pathways for Rett syndrome pathogenesis.
Hello and good evening, and I want to thank the organizers for allowing me to speak, but I'm one of the organizers. So anyway, so tonight I'm going to tell you a bit about Rett syndrome. It is an X-linked neurological disorder that's caused by mutations in an epigenetic factor called methyl-CpG binding protein 2. Now, Rett syndrome is a rather devastating disease because girls are usually born normally and they develop normally, and then they go through a developmental regression where they lose their ability to speak, they lose acquired motor skills and withdraw socially. They have autistic traits such as stereotypies, such as tapping their hands. They also can develop things like abnormal breathing, seizures, and most of them have GI problems. Now, Mecp2 is prevalent and central to neural development. In fact, boys with severe mutations die at birth due to encephalopathies, and milder mutations in Mecp2 are associated with intellectual disabilities and autism. So Mecp2 is very difficult to study. It has been reported to both repress and activate genes in DNA. It has also been reported to bind RNAs, and so you can imagine that it influences many, many different pathways. And it's very difficult to sort out which ones are important for symptom progression. Although the disease was first found by Andreas Rett in 1966, and my colleague at Baylor College of Medicine, who cloned the gene in 1999, mouse models that were developed around 2011 were very informative for the disease, but really we've made little progress in understanding why the symptoms occur. So we thought we could use mouse models to find mutations in unknown genes that might improve the pathological phenotype caused by Mecp2 mutation. And I put this timeline on here to let you know that we carried out our initial round of screening for mutations in 2 years. It then took us about 2 years to work out mechanism, almost 2 years to get the paper published, and then the screen started up again in 2013. And this is just so you students don't get too depressed. But anyway, we decided that the best way to tackle Rett syndrome was really a forward genetic modifier screen. So in such a screen, suppressors and enhancers of the phenotype could pinpoint pathways. We felt that this was the time to do it because we had next generation high throughput, low cost sequencing, which would make mutation identification simple, especially on inbred strain backgrounds. And therefore, we could identify genes with functional significance for Mecp2, but some could be alternative targets for therapy. So our screen was a dominant suppressor screen using the chemical mutagen ethylnitrosourea. We mated wild-type males with Mecp2 females, and we looked at only males. We genotyped them, and those males that carried the Mecp2 mutation were evaluated for health traits and symptoms that included limb clasping, tremors, muscle tone, activity, and body weight. We scored them so that would equal a health score. These males would normally die by 6 to 12 weeks of age, and we were looking for an ENU-induced mutation that would improve symptoms and therefore improve the health score and give them perhaps increased longevity. So at this point, we have screened over 5,700 male mice, and that is, by the way, more than 11,000 mice. We have isolated 58 lines that show suppression of traits, and these lines improve different phenotypes, suggesting that we're probably isolating different genes. We carried out whole exome sequencing to identify the lesions and have identified lesions now in 46 of the lines. These lesions represent 32 candidate genes because 7 genes have independent repeat hits. In fact, one gene has 3 different hits. 3 lines carried 2 independent suppressors that when they are together improve traits better than either one separately. And most excitingly, the mutations are lying in components of common pathways and co-repressor complexes. And I'll give you an example of that near the end. But first, I want to tell you about one very exciting allele called suppressor of Mecp2 number 3. And this was a line that showed extremely good suppression of traits. In fact, the original founder male for this line lived to 14 months of age, and we had to sacrifice him. This line turned out to be a null allele of a gene called Squalene, epoxidase, and it was identified by both linkage mapping and whole exome sequencing, but turned out to be a complete loss of function allele. Well, this was very interesting because SQLE is the rate-limiting enzyme in committed cholesterol synthesis, although cholesterol synthesis had never been associated with Rett syndrome before. To make a long story short, I'm going to tell you years of work and 2 papers worth of data, but we found essentially that lipid dysregulation was a systemic problem in Mecp2mice. So brain cholesterol has to be produced in situ because it cannot cross the blood-brain barrier. Old cholesterol in the brain becomes damaged and has to turn over for appropriate neural function. So Mecp2mice produce high levels of a neuron-specific enzyme called CYP46A1 that hydroxylates cholesterol to allow it to pass one way back through the blood-brain barrier. This causes a cholesterol buildup that then shuts down synthesis in the brain and is not a good situation for the mice. But we also found, to our surprise, that systemic lipids were also overproduced, and this led to fatty liver. It caused high insulin levels and insulin resistance in both males and females and led to metabolic syndrome. And we have now published a paper suggesting that Rett syndrome be considered a neurological disorder with metabolic components. But we really wanted to know how Mecp2 could deregulate cholesterol synthesis. Well, a clue to this came when Adrian Bird published that Mecp2 is required to bridge the NCOR1 HDAC3 repressor complex with DNA. Of course, he reported that that was true in neurons. But we were very excited about this because the type 1 histone deacetylase HDAC3 is a master regulator of lipogenesis. In fact, if you are inactive in feeding, you are going to suppress your lipogenesis genes, but if you're active in feeding and perhaps eating too much, you're going to turn them on. And this happens in a diurnal fashion as we go through our daily periods. So we thought then that perhaps Mecp2 could interact with HDAC3 to regulate lipogenesis. And we chose to look at this in the liver because the liver is a rather homogeneous organ, unlike the brain, although we thought it would give us clues to what was happening in the brain. So my graduate student, Stephanie Kyle, first carried out a co-immunoprecipitation using HDAC3 as our antibody and found that indeed we could pull down Mecp2 in wild type, but not in the null. And NCOR1 was pulled down no matter what. She also carried out chip sequencing using H3K27 acetylation, which is a downstream target of HDAC3. And I'm showing you here the profiles for wild type and Mecp2 in blue at 3 different genes: SQLE, which is responsible for committed cholesterol synthesis, CD36, which is responsible for lipid uptake, and fatty acid synthase, which is responsible for lipid synthesis. And you can see that in all of these loci, in the absence of Mecp2, we had increased H3K27 acetylation. She also carried out CHIP qPCR to assess HDAC3 binding in the absence of Mecp2 at SQLE. And she chose 3 different sites in order to do this. And you can see that HDAC3 bound SQLE less well in the absence of Mecp2. So we concluded that SQLE is a direct target of this complex in the liver and probably in the brain as well. Now, so I've shown you this very interesting complex that would normally turn off SQLE at appropriate times. So when Mecp2 is absent, you would expect SQLE to be turned on constantly. And indeed, we have found that a mutation in SQLE that downregulates the gene improves symptoms, but we have also found missense mutations in two different complexes, two different components of this complex that regulates SQLE. We've also found mutations in other targets of this complex, including acyl-acetyl-CoA synthetase, and in hexokinase 2, we found two different mutations. And so we think that actually targeting various components of this pathway will improve symptoms in these mice. Now, I've shown you the suppressor and the pathway, but this also begs the question that FDA-approved statin drugs could be repurposed to improve symptoms. And indeed, we used a lot of different testing protocols on both males and females to see if symptoms were improved. We found that intermittent or low doses greatly improved both male and female symptoms. Interestingly, a daily dose of statins was not as effective, and trying to reverse lipid buildup in aged 6-month-old females was also not effective. So essentially, the statins would prevent the lipid buildup and the shutdown in both the brain and the liver. But we're also trying a number of other drugs developed for type 2 diabetes that may show promise for this disorder. So we really want to know if these data will translate to Rett patients. Our predictions from our mouse work is that a subset of patients will have peripheral dyslipidemia. This is because many different genes modify this pathway. Some may be also prone to liver disease. We think that brain cholesterol homeostasis will be dysregulated in all patients at some point, but it will vary with disease progression and is difficult to assess. But we think that 24S-hydroxycholesterol and insulin might be good biomarkers. Clinical trials are now underway in both New York City and Italy. So I think I've shown you that modifier screens can reveal disease pathology downstream of transcriptional molecular events. It's the time to do this now with next-gen sequencing. Our suppressor screen is ongoing now to find all therapeutic targets for this disorder. We now know that HDAC3 requires Mecp2 for regulation of SQLE, and we think that this complex also regulates it in different cells at different times in the brain. Genetics can inform therapeutics. Our incomplete rescue for each allele suggests that combination therapy will be required, and that is also supported by the idea that two different suppressors in different pathways put together will actually improve symptoms even more. So our studies of Mecp2, I think, will inform other neurological diseases. I wasn't convinced that this was a good target when we first started, but it turns out to be a great target. And now we think that absence of Mecp2 actually puts the genome out of balance, and it's very easy to tip the scale one way or another. And so it's been very productive. This work required a lot of people, but it was primarily led by two very talented graduate students, Stephanie Kyle and Christy Bukovecki, in my lab. And I'll stop there and put a plug in for my journal, Disease Models and Mechanisms, and I'll take questions.
In the interest of time, let's have one question or two brief questions for Monica.
With MECP2 being able to now bind to CPA methylation, can you comment on the relationship between K27 acetylation and CPA methylation?
I'm not sure I want to comment on that right now, but I do think, yes, that there are probably links, especially now that Adrian has found that the major thing that Mecp2 binds are actually CPAs or other derivatives and not necessarily just CPGs.
So you alluded to the fact that a lot of the suppressors were in NCO, well, in related epigenetic regulators. And I just wonder how you think that these things can, I mean, it's one thing to have one regulator, a point mutation in MECP2, affecting a subset of genes in the nervous system. But it's another thing to think that the entire pathway could then collapse upon those sets of genes where these are very ubiquitous regulators. So, I mean, the mice must have a lot of other phenotypes as well, or is it highly specific?
Oh, they have lots of other phenotypes. What's very interesting is that when you can get them to live longer, they develop phenotypes that we didn't see in the ones that die earlier. I should say that the ones that fall into this pathway, I didn't tell you all the pathways, tend to have very similar phenotypes. However, we can almost tell which traits are rescued and predict that it will lie in that pathway now. But I must tell you that these mutations, for instance, in COR1, which would affect tons of things, is a missense mutation in a very special region of the protein. And actually, these missense mutations are telling us a lot about the function of those particular components.
And so what percentage of the suppressors were these sort of global regulators, and how many were more specific downstream targets?
How many? I would say that probably over 50% are predicted to affect chromatin.
Let's thank Monica again.